My name is Zach. I am the Hemp Curator in Geneva, New York. I'm also the acting Vegetable Crop Curator, so I manage the tomato, brassica, and many other crop collections. I saw some of the coloreds I keep in Nico's wonderful talk, but today we're here to talk about hemp. So I wanted to do a very quick overview of hemp and what sorts of work we are doing at PGRU to conserve, characterize, distribute, and now breed it. So Cannabis sativa is one species, despite what you may hear in the media about different indica sativas. Those things are not true. There's a lot of population stratification in hemp, but they do not follow those labels. Almost everything I'll say about hemp could have an asterisk. Hemp is typically dioecious, but not always. There are monoecious cultivars. Hemp is a diploid, usually with 10 chromosomes, although tetraploid and even triploid varieties exist. It tends to be a short day crop with strong day length sensitivity to trigger flowering, although day length insensitive varieties exist, and it is used for many, many different purposes. Our work at Geneva is kind of threefold. We want to simply understand hemp germplasm research, breeding, and conservation efforts. I tell people that, that the challenging aspect of working with hemp is we don't have a deep body of literature like we would in many agronomic crops. However, that's also really exciting because there's so much new soil to turn over in hemp science. Secondly, documenting, preserving, distributing, and evaluating all of the resources that we are just now really beginning to gather together. And then maybe even taking this tabula rasa opportunity to develop and apply novel genomic approaches and apply them to hemp itself. So this is a quick and dirty little map that I made where I scraped a bunch of different online resources, including GBIF, which we were talking about earlier, the old USDA collection, and just different herbaria samples from all over the world. And my point is that hemp is globally distributed. It's been in human use for at least 6,000 years. And it's become widely adapted to many different environments and conditions for many different human uses. So hemp is no stranger to the plant germplasm system. This is a scan of plant introductions. The first hemp was PI 948, 3-digit PI numbers. This one was most likely part of the Nikolai Vavilov collection that was transferred to the United States in 1898. So one of the earliest efforts that we made in this process of understanding hemp and the needs of our stakeholders and customers was to ask them a lot of questions. We conducted a USDA stakeholder survey with help from our friends at Cornell University, and got nearly 800 responses to this. And the answer kind of collapses to everyone is interested in almost everything about hemp. Over half of the respondents, rated these particular phenotypes as moderately to strongly important to understand when using PGS germplasm. But unsurprisingly, cannabinoids hold the greatest interest for many reasons, notwithstanding legal implications. So just a quick slide deck in here of the updates. Probably the most important change is this young man, Tyler Gordon, who has been hired as our hemp breeder. He's a ARS as a small grains guy, and now he's in the East Coast working with hemp. And we're just absolutely thrilled to have him. He's already started so many incredible projects. We've collected about 188 sessions up to this point from various public, private, university level groups, breeding programs that clearly wouldn't donate their elite germplasm, but they may have breeding stock or dead end breeding lines that they've been able to turn over. And we are also working with University of Wisconsin to comprehensively gather feral populations from all over the Midwest. So we've begun increasing these materials. In many cases, we had 100 seeds. So increase is really a critical first step for most of these populations we received. Tyler and I have started a large field trial with everything we had sufficient seeds of this year, and we are evaluating these accessions for likely up to about 40 different phenotypic traits. As sort of a precursor to that, we developed a hemp phenotyping handbook and presented it to the New Crops Germplasm Committee, and they approved that. We've also set up some really powerful phenotyping collaborators inside and outside of the ARS. And we're thinking very carefully about developing a common genotyping platform for hemp. This is a surprisingly complex equation to solve, but this is something where we really would like to get right. So this is just a quick look at the phenotyping handbook that I wrote with my close friend Anya. So we combed through about 400 different journal articles, trade publications. Anya is a Russian speaker, so she translated a lot of Russian hemp texts. We also interviewed a lot of people, asked for their opinions, made a Google document, sorted things out by phenotypic class, such as leaf, seed, fiber, and fluorescences. And we started some fights in the Google document, which was good because that shows us where people disagree or where we could improve our phenotypic evaluation. So this is an example of a morphometric analysis of hemp architecture done by my friend Craig Carlson. So this is a way that you can apply yardstick phenotyping to a hemp plant, but it's also highly amenable to high throughput drone or robotics phenotyping down the road. So we're trying to develop these phenotypes that are understandable to humans, but also are amenable to greatly upscaled robotic type phenotyping efforts. This is some of our phenotyping collaborators. So Mark Burhow at NCAUR is evaluating all of our materials for cannabinoids and other secondary metabolites, so terpenes, phenolics, and others. Chris Delhomme at the Southern Regional Research Center is a fiber guy. He's worked a lot with cotton, but also flax, which is also bast fiber like hemp. So retrofitting those tools to evaluate things like hemp fiber quality, ease of decortication, et cetera, are going to be really essential in the next 10 years. We're lucky to work with Dr. Siebert at Alabama A&M, who is helping us on a lot of different tasks, including morphological phenotyping or plant architecture studies. And then we're working closely with Cornell, UC Davis, Oregon State, Wisconsin, just trying out a lot of the phenotyping platforms and stuff that they're developing or otherwise exchanging information and tools. All right, so just a quick overview. Hemp can be male, female, or monoecious. It has an XY chromosome system, although it gets very confusing very quickly. we are beginning to gain some glimpses into the underlying population structure within the wider pool of hemp genetics. This is a really nice GBS study done by the same Craig Carlson, and he found that really a lot of things, but there's some major population differences between fiber, grain, and CBD cultivars, and they segregate into pretty distinct groups. So how do we build our collection to capture that wider pool of genetic diversity? So this is one I get all the time. People say, what about marijuana, or what if it goes hot? And so I break out biochemistry, and they usually regret asking me that. However, I'm going to wildly oversimplify cannabinoid biosynthesis. Biosynthesis of THC and CBD is largely controlled by one locus we call the B locus. They're not strictly speaking alleles, but they can be readily modeled as such. THC and CBD are decarboxylated from a THCA and CBDA respectively, and those molecules are produced in turn from CBGA, which is a precursor for which THCA and CBDA compete for. Some fantastic work by Dr. Jacob Toth at Cornell has really delved into these BD and BT alleles and shown how they can segregate in a Mendelian fashion, producing these three chemotypes that are defined phenotypically by their CBD to THC ratio. So chemotype 1 is strongly THC dominant, chemotype 3 is CBD dominant, and chemotype 2 is a heterozygote and displays an intermediate phenotype. So Jacob even developed these fantastic pace markers for this B locus. So we can see the three chemotypes here, and in fact screen a plant at the first true leaf stage and get a very accurate prediction of its final total potential THC concentration. It's also worth noting that Jacob has drawn a red line here at 0.3% total THC. That's the legal limit. So simply having a BD genotype is not sufficient to ensure regulatory compliance. As you breed for progressively higher and higher CBD, you will cross that 0.3% THC line around 7% CBD. So this is sort of a glass ceiling for CBD producers right now. The regeneration question is another really, really tough one. So working with these wonderful vegetable crops, our standard regeneration uses growing an accession in a nylon cage, releasing a pollinator, and if all goes well, collecting seeds in the fall. This really doesn't work at all with hemp. Hemp is wind pollinated. Hemp pollen grains are about 20 microns in diameter, and you have an expectation that hemp pollen can travel 5 kilometers by wind. So pollen control is absolutely vital to maintain the genetic purity of an accession. And it really is a challenging endeavor to regenerate hundreds or not thousands of hemp accessions with this incredibly tight pollination control technique. So these are some of the approaches that we're kind of trialing or running right now. So going clockwise from the top left, we have growth chambers, pollination bags, and greenhouses. These special pollination tents with 10 micron pore size, and simply growing one population at a time in a greenhouse. This is hard, very time consuming and expensive. We've developed a couple of stopgaps in the meantime. So collaborations, people with unused greenhouse or growth chamber space, like Dr. Siebert at Alabama A&M, can help us regenerate these populations as quickly as possible. We're also developing in-house some positive pressure systems to essentially subdivide our greenhouses into smaller pollen-controlled greenhouses, as well as even developing field cages with 10-micron mesh. So I really want to highlight the work of Dr. Nicholas Jenna, my postdoc. He is a seed scientist, which I understand are kind of becoming rare these days, but we're so thrilled to have him. So he's been really drilling into this hemp seed question. Involves a lot of different factors like dormancy and irregular germination in the field when viability tests indicate entirely viable seeds. So here is a pollen collection device that we've been developing in-house. So clockwise from top left, here's an image of hemp pollen. Nick has been evaluating different TZ testing to determine pollen viability. We have developed this device on the bottom left here, which is a tube with a series of progressively smaller filters down to 10 microns that we can attach to a shop vac and effectively vacuum pollen off of male plants and produce large quantities very quickly. Nick is doing a lot of work comparing pollen viability in different treatment regimes under different drying conditions, et cetera. And he's in fact been able to get pollen to germinate on Brubacher clock media and is learning a lot of things about drying, over drying, and we hope to work with more NPGS folks to dig deeper into this problem. So pollen is a really good stopgap to distribute alleles to the breeding community while we are trying to produce sufficient seeds. So we feel that developing good pollen collection, storage, and distribution protocols would alleviate some of the breeding logjam that's occurring with hemp. I also want to give a big shout out to Tony Baracco, who is working with me. Tony is studying fiber quality. Hemp undergoes a rutting process, which is typically a type of decomposition that happens in the field, and studying what phytochemicals degrade, what persist, and studying that across cultivars. And we're working closely with Christelle Holm at the ARS lab in New Orleans. So how do we develop fiber quality grades for hemp like you might have for cotton, for example? It's another bedeviling question. So I just want to thank really everyone that has contributed to this project. This isn't even an exhaustive list, but I've tried to put everyone that's been contributing. This is our field that we just planted at the beginning of June. And yeah, sorry, that's a lot of information all at once, but thank you for your time. And I really want to thank Claire, Brian, and Jill for putting this together. It's been a great workshop.